The overall goal of this procedure is to assess granulocyte function as a two-parameter approach. This is accomplished by first incubating granulocytes from patient blood samples with bioparticles and a reagent to visualize the oxidative burst. In the second step, the post-incubation phagocytosis is blocked, and then the cell surface receptors of interest are labeled to allow for positive identification of the granulocytes. Ultimately, the activated granulocyte subsets can be identified and isolated by image-based flow cytometry.
This method can help answer key questions in the nutritional or clinical immunology fields, such as how dietary habits and nutritional practices contribute to alterations in granulocyte function. Demonstrating this procedure will be Eric Prado, a doctoral student from my laboratory.
Upon obtaining the peripheral blood sample, thaw the S. aureus bioparticles and DHE at room temperature and N-ethyl malamide in a 37 degrees Celsius bead bath. When the reagents have thawed, add 20 microliters of the S. aureus bioparticles to 4 individual 1.2 milliliter tubes within a sterile hood. Then add 40 microliters of the thawed DHE to each tube containing the bioparticles and gently tap the tubes on the bench to collect the reagent in the bottoms of the tubes. After adding 100 microliters of mixed whole blood to each tube, remove any contaminating blood along the inside edge of the tubes with a cotton-tipped applicator and mix the blood and reagents with an electronic pipette set for 3 cycles. After the 3rd cycle, place all of the tubes in an ice bucket protected from light. Then incubate the tubes for 10, 20, or 40 minutes in a 37 degrees Celsius bead bath, starting with the 40-minute tube.